All right, so this second part of sensory physiology is going to deal with a little bit more complex of the sensory system. We're going to be looking at what's taking place in the ear and the eye. So with the ear, we're going to get auditory. We're going to get sound. All right. We're also going to get equilibrium and balance off of the ear. So both of those senses are coming from the sensory apparatuses in the ear, and then in the eye we'll be looking at basically back at the retina, where we'll find those photoreceptors in which we get vision information. So hopefully this looks a bit familiar from anatomy, but in that inner ear, in that temporal lobe, you have this bony labyrinth. Okay? And so the sensory structure located in, we have the bony labyrinth, okay? And perilymph lies between the bone and what we call the membranous labyrinth. So the membranous labyrinth basically follows the same bony labyrinth, it's just an inner structure, and that membranous labyrinth is going to contain endolymph fluid. Okay, so on the outer edges, or the outer side, up between the bony and the membranous labyrinth, is perilymph. Perilymph is very similar to interstitial fluid. We're going to see endolymph, that inner, basically flanks inside of the bony labyrinth, is high in potassium. Okay, so the sensory cells that are going to be located in this membranous labyrinth are bathed in a fluid that is high in potassium. So keep this in mind, because say here's our sensory cell and we have potassium high outside of it, right? Usually you have potassium high inside the cell. What's going to happen if you had a channel that opened for potassium? In this case, because it is bathed in this endolymph, potassium would rush in and cause depolarization. All right, so keep this in mind as we go through and looking at these structures of the inner ear. So part of that inner ear is going to contain what we call the vestibular apparatus. Here is where we're going to get equilibrium, how we're going to get motion sensed. Okay, and we have two structures making up this. We have basically the vestibule with the utricle and sacula. Okay, and these are going to give us linear acceleration. So acceleration in one plane. So think of a plane going forward, backwards, and up, down. Okay, forward, backwards, up, down. Okay, looking here, utricle example is, if anybody recognizes this, this is the Montezuma's Revenge at Knott's Berry Farm, but you take off in this very fast acceleration, okay, horizontally. Eventually you go vertical, but this very first stretch you're going horizontally. Okay, that would be the sensory receptors of the utricle. They're set up in a line that when we go horizontal acceleration, we're going to get bending of these sensory receptors. The opposite, scapula, gives us the up-down motion. Okay, again in one plane, just up-down. It's not a circular motion, it is up-down, one plane. Okay, and that is the sacula. Okay, when you're jumping rope, you're going up and down, up and down, up and down. And the sacula, the sensory cells of the sacula, are going to be able to be stimulated by that vertical acceleration. Okay. Then there are semicircular canals. We have 3 of them located part of this vestibular apparatus, and it's going to give us dynamic acceleration or rotational acceleration. Okay, these are the semicircular canals. When you do a pirouette, you are activating those semicircular canals, and those are the ones that are picking up, hey, I'm moving in this circular plane. All right, and we can pick up the motion to give you equilibrium so you just don't fall down. So let's look at the sensory cells of the vestibular apparatus. Again, these are modified epithelial. Okay, so they are not neurocells, but they're going to be associated with the neurocells, and these modified epithelial have this microvilli, or what we call stereocilia. Okay, these hair-like projections. Okay, stereocilia, and on the one end we have what's called the kinocilium, the larger. Okay, but they are tethered together, so when these move, the kinocilium is going to be pulled with them. Okay, depending on which direction the movement's taking place, we're going to see movement. Okay, and so it is the bending. So these are mechanoreceptors because it is the bending of the membrane that is going to open the channels and cause either stimulation or inhibition. Okay. The kinocilium. Okay. You can see here, let's see what's happening to action potentials when we do get bending of these sensory cells. Okay. And you can kind of see this arrangement here. It's kind of cool seeing the smaller side here, okay, here, and then the kinocilium up over on that end. But bending, you can see bending in direction towards the kinocilium. Okay, so if this is bathed in fluid, you got to imagine that fluid is running past here, okay, that endolymph, and it would cause bending of these sensory cells, okay, and bending of these kinociliums, all right, or stereocilia with the kinocilium. When we bend in the direction toward the kinocilium, you can see the frequency of action potentials of the sensory receptor that are associated with the sensory cell, very high frequency. When we bend in the opposite direction, away from the kinocilium, okay, you can see we get inhibition. Okay, and then when they're standing straight up, you can see there's always a baseline for these sensory receptors. The brain's getting a baseline, and what the brain's doing is putting an interpretation. Hey, I got a baseline, there's no movement. Hey, I got high frequency, I'm moving in one direction. If I get inhibited by the sensory neuron is inhibited, I'm getting this inhibition from that sensory neuron, I can say I'm moving in the other direction. Okay, so that is how the sensory receptors, these sensory cells, are basically putting perception to movement. And we can see the movement is going to be taking place due to fluid moving across here. Fluid moving in one direction or the other direction is going to bend the kinocilium in either way. Looking at the utricle and saccula. Again, this is linear, either up, down, forward, back. Okay, motion. Okay. Forward, back, utricle, up, down, saccula. But basically, here's how those sensory cells, okay, those kinocilium, looking here, they are actually embedded in this membrane. Okay, this otolithic membrane with these otoliths, these crystals in here, okay, of calcium carbonate, gives it a higher mass. Okay, and you can see the utricle and saccula. The utricle is basically in a horizontal plane because it's giving us horizontal, okay, acceleration. So if I was to move, here's the anterior, if I was to move in it horizontally anterior, okay, these are going to be pulled backwards. And the saccula is arranged in a vertical type direction. So if I jump, okay, it's going to be pulled down, and then I get changing of the bending of these kinocilium. And you can see when the head is in right plane. Now I move in one direction. Here's bent toward the kinocilium. I get increased. All right. And now I bend in the other direction. If I'm looking at this kinocilium, I would be getting inhibition. You can see there will be others that are arranged in the opposite direction. The kinocilium would be on this side. And so basically the brain's getting multiple information. It get information from a sensory cell that's bending towards the kinocilium and getting information from these located in a different region that would be inhibited. So the brain can put this information together. But basically it is the bending of the stereocili pulling along with the kinocilium that is going to give us the signaling for the receptors. So now let's look at the semicircular canals, and you can see there are three here. We have the superior, running in one plane, the lateral, and then the posterior. You can see their arrangement is almost 90 degrees to each other, so they're going to get different movement. But what's going to happen is as you move in a rotational type way, the fluid, the endolymph in here, is going to move with it. Okay, and so the endolymph is in here, and the sensory cells, again, kinocilium with stereocili, these cells are located in the crista ampullaris, and these hair bundles with the kinociliums are projected up into this cupula, this gelatinous structure. Okay, so as the endolymph moves across, it can bend this cupula and in turn bend the kinocilium and the stereocili. Okay, so we get the bending through the movement of the endolymph, or lack of movement, the inertia of the endolymph. We will see here in a sec. Okay, but the endolymph will cause the bending of the cupula, and then we can get the bending of the cells, and we get stimulate these hair cells located in the crista ampullaris. So the movement, you can see we're doing a little pirouette here as she's moving rotationally. Okay, what happens is the inertia. The fluid doesn't move as fast as the actual cupula. Okay, so the fluid stays still. This cup is moving, but the fluid staying still, and the inertia actually pulls the cup in the opposite direction of the movement because of the slow movement of the fluid to respond. Okay, after we continue rotating, the fluid's going to start going in the opposite direction with the movement. It's going to start rotating around. Okay, and then when you go to stop, you're slowing down. The cupula is going to get pulled in the opposite direction. Okay, because of the acceleration. It's not velocity. If the velocity is the same, if we're going at the same rate, you're not pulling on this because the fluid is going at the same rate as the actual cupula, the semicircular canal. But when you first go to move, you get acceleration, you get the inertia, and then as the fluid's moving and you go to slow down, again, you get pulling on the cupula and you get signaling of those hair cells. You get depolarization. Okay. What channels do you think are open when you're getting bending of these hair cells? What ion channels? Yeah, in this case it's potassium, because remember, this is endolymph that is flowing through here. Endolymph is high in potassium. Okay, so with our neuron, if we had potassium channels open, potassium's flowing out, but because the endolymph is high in potassium, potassium is flowing into the cells. Okay. And you can see the arrangement of these semicircular canals. We can get different movements. You think the superior. Okay, it's basically, if you put this would be lined up, this would be the front of the head, this would be the back. That is kind of the lineup. And so when you do a somersault, you're flipping over, that would be stimulating the superior. The lateral. Okay, almost like a left-right plane. Okay, horizontal plane. And so when you do a spin on the long axis, like she's doing here, this pirouette, that would rotate the fluid in this semicircular canal. Okay, and then a cartwheel. You see the cartwheel is basically lined up vertically to the left-right direction. And if you do a cartwheel, that would spin the fluid or move the fluid in this semicircular canal. Okay, so we can get different planes of movement with the different canals. Some movements may be partial lateral, partial superior. Okay, depends on the movement and what's going on. Okay, but the brain's getting that information and it can recognize, hey, where's the body, where's the movement of the body taking place, and can keep you in equilibrium, keep you from falling, basically giving you that balance. So here's basically the bending of the cells, right? Pressure wave on the stereocili, the cupula is bending. Okay. And the bending in this case, because we're bathed in endolymph, we're high in potassium here. The bending towards the kinocilium opens up potassium channels. And in this case, because potassium channels are open, potassium rushes in and we get depolarization. And just like a neuron, remember this is modified epithelial, but it's going to signal similar to a neuron. Depolarization causes calcium to flow in, and then the vesicles fuse and we get neurotransmitter released to the afferent or the sensory neuron. That brings that information up to the brain. And so the neural pathways for this equilibrium, your eyes are part of that too. Okay, it's not just this vestibular apparatus. This gives you one piece of the information. Okay, you also have those proprioceptors telling you where your joints are. Okay, and so your brain's getting information from the eyes, from the vestibular apparatus, and from those proprioceptors in the joints and in the muscles, those muscle spindles. Okay, and that information comes together at the brainstem to give you a sense of balance, to allow you to move in these movements and keep you from falling down, give you that balance. Okay, so if you ever try balancing on one ankle, right, with your eyes open, it's much easier. And then close your eyes, and what happens? You lose this portion of it, and so you're getting some of the vestibular apparatus, but getting some of the proprioception. You're losing the eye portion, okay? Or doing a pirouette with your eyes closed. You get the vestibular apparatus and the joints, but not the eyes. You see, you're missing part of that information. You're not getting the full picture, and it is much harder to complete that movement without falling. Okay, so the vestibular apparatus gives you part of the equation, but you also have these other parts that help you stay within balance. So now we're going to move on to the second piece of information we get out of the inner ear, and that is auditory, hearing. Okay, and we saw the vestibular apparatus, we had vestibule and the semicircular canals, and that snail-like structure when we looked at the bony labyrinth earlier contains the cochlea. Okay, that cochlea is where we're going to have hearing. Okay, and what is hearing? What stimulates these cells is sound waves. Okay, and we can get two major pieces of information that we're going to be used to obtain auditory information, and that is the frequency. Okay, how frequent are the waves? Okay, if I drew this is a much frequenter than, say, down here, low frequency. Okay. The other piece of information is I can get intensity. And intensity happens with amplitude. Okay, high amplitude, low amplitude. Okay, so the height, right, and the frequency are going to give us two different pieces of information. The frequency is going to be with our pitch, and then the intensity or the amplitude is going to give us our intensity, how loud the sound is. And usually measured in decibels. Likely you want to keep it between 0 and 80. But if you go to a rock concert, you're going to be well above this area. Okay, so the amplitude is going to be much greater with a rock concert. Okay, but that's going to give us our two pieces of information, and this is what's going to stimulate our cells, is these sound waves coming into the ear and ultimately moving in to that endolymph fluid in the cochlea. So for hearing, there's going to be three different structures of the ear. We're going to have the outer ear, the middle ear, and the inner ear, where we have the cochlea with the sensory cells. Okay, but what we're going to have is we're going to have sound waves coming in, right? And the outer ear's job basically is to funnel the auricle, the ear, to funnel the sounds into the external acoustic meatus and ultimately onto the tympanic membrane, okay, or the eardrum. Okay, and basically that is the sound waves coming in and hitting the eardrum is going to cause vibrations on the tympanic membrane. Okay, those vibrations are going to be basically amplified in the inner ear by the ossicles. So let's take a look at the middle ear here. Okay, so the middle ear between the tympanic membrane and the oval window of the cochlea. The cochlea is where we're going to find the sensory cells. Right now we're just moving the sound waves in. Okay, so sound waves are coming in and hitting this tympanic membrane and causing vibrations on it. That's going to move these ossicles. Okay, you can see kind of like the alien baby here climbing up the tympanic membrane. Right, that's the malleus, the incus, and then the stapes. The stapes is what connects to the oval window here, and the oval window is basically the entrance in to the cochlea. But basically the function of the tympanic membrane is, if low sounds, basically to amplify. We're going from this big tympanic membrane and we're causing movements here and pushing into the endolymph fluid that is in the cochlea, okay, onto that oval window. So we're going from a big window to a small window, so we're getting amplification of the sound waves through the middle ear here. So in the inner ear, the cochlea, okay, this is where we're going to have the endolymph. You can see here, there's that little oval window that's pushing in, and that stapes is pushing into the endolymph fluid in this cochlea. You see the cochlea makes these three turns. So it goes around. Okay. And inside this cochlea, we actually have three canals. Where the oval window is pushing into is into the scala of the vestibuli. Okay, the scala of the vestibuli, part of the bony labyrinth, and so it has the perilymph. Okay, so scala of the vestibuli. Okay. And then the other part, so this is continuous. As we roll in, okay, the fluid rolls in and it takes three turns. You can see it kind of rolls around this way, and it'll roll up here, and roll up there. Okay, and then it would continue into the scala tympani. So it is continuous. It's just the back end is the tympani, and that's this portion here. Okay, and again it is perilymph. Wedged in between these two is the membranous labyrinth with the endolymph, and that is making up the cochlear duct. Right, this little structure here. or let's use here, is the cochlear duct, and we can see this is where the sensory cells are going to lie. Okay, so this is basically, it would be the beginning end where the fluid gets pushed in, right? And we get these sound waves brought into, and those sound waves move up, right, making these turns. Okay, so the sound waves are first brought in the scala vestibuli, and we're going to see that is going to cause movement in the cochlear duct. Okay. All right, let's look at this cochlear duct, or also called the organ of Corti, or spiral organ, that is going to contain. Right, we get the scala vestibuli. That is where the sound waves are brought in to, so we get vibrations through the fluid, perilymph fluid in the scala vestibuli. So the oval window is connected to the scala vestibuli, and so the sound waves that are coming in from the middle ear are brought into the scala vestibuli, and you can see they make those three spirals, and then they're continuous with the scala tympani. Okay, in between again is that spiral organ, or organ of Corti. And it's going to have this tectoral membrane here. So let's take a look. This is actually blowing up down here. Okay, you can see there's this basilar membrane. This would be the scala of the tympani up here. Okay, and the tectoral membrane is here. And above here is endolymph. Okay, so we have that high potassium fluid here. Okay. Attached to the basilar membrane are these outer hair cells and these inner hair cells. The actual sensory organs are the inner hair cells, and there will be a row of them across here, and there will be 3 rows of these outer hair cells. We'll see what their function is, but the actual sensory cells that are going to respond to the sound waves, to the vibrations, are going to be the inner hair cells. Okay, and they are embedded in this tectoral membrane. So if we have sound waves coming in that are bouncing across here, that are causing movement in this tectoral membrane, we're going to get bending of the hair cells here, the inner hair cells. Okay, as we saw, we got the stereocili. In this case, there is no kinocilia like we saw in the vestibular apparatus, but there are those stereocili embedded in here. And if we get sound waves brought in, causing shearing force, these tectoral membrane moves across, it's going to bend these stereocili at. So as those sound waves are moving in, right, they're going to move into the scala vestibuli, and they're going to be translated into the basilar membrane in which those hair cells are embedded. Okay, and those stereocili are embedded in that tectoral membrane, and what happens as this membrane gets moved up and forward, there's going to be a force causing the bending of the tectoral membrane. Okay, a shearing force. Okay, depending on how the bending is taking place. Okay, but that bending is going to bend those stereocili, and again we have potassium channels. When these get bended, we have potassium channels opening, and this here is endolymph fluid, which is high in potassium. So again, just as we saw with the vestibular membrane, but no kinocilia here, but the bending of the hair cells, or the stereocili, end up becoming depolarization by opening of potassium channels. Okay, so here's the sound waves coming in, bending, causing vibrations on the basilar membrane, causing that tectoral membrane to shear and bend these stereocili, and now we're going to open up potassium channels. Okay. That's potassium comes in, depolarizes the cell, causing calcium influx, and we get depolarization and activation, neurotransmitter release to the auditory neuron, and then we get the auditory neuron signaling, bring the signal up to the brain. Okay, so again, these are modified epithelial. They're not neurons, but they act like neurons in that they get depolarized and they will release neurotransmitter to the sensory neuron to get stimulated. Now let's see how strength of stimulus is coded in these hair cells. The loudness. All right. Say I talk like this, and I'm sure this worked better in class because I jolted you guys up in class, but I might have an amplitude of this. And then when I talk real quietly, all right, I might have an amplitude such as this height. Okay, so bigger amplitude with more loud, and softer, I'm going to have a lower amplitude. You can imagine what's happening to the bending, because remember these sound waves are brought in to that perilymph and they cause distortion of the basilar membrane. Which one's going to cause more distortion and probably more bending of the hair cells? Yeah, the loud, the more higher amplitude. So let's say this was for the quiet here. You can see, all right, no bending. That hi, how's it going? All right, the quietness going to cause some bending, and I'm going to get frequency. But how about the loud? The loud's going to cause even, say, higher bending of those cells. Okay, greater distortion, and you can imagine we're going to have very, very frequent action potentials with the loud. So loudness is how frequent, or the frequency of action potentials by the sensory neurons. Okay, if I get more bending, I'm going to have more neurotransmitter released, and that's going to cause me to have a higher frequency of action potential for the loud. Okay, so strength of stimulus is going to be coded by frequency. Now the other characteristic I get from auditory is pitch. Hi, how are you doing? Very high pitch sound. Hi, how are you doing? Very low pitch. Okay, so higher frequency. Higher frequency is going to be much more frequent, or low frequency. Okay, and which one do you think you're going to get distortion? Remember, there's that spiral. We go in 3 circles. Okay, 3 turns for the cochlea. Say this is the beginning, and this is the end up here. Which of these do you think would stimulate the cells earlier on in the cochlea, and which one do you think will stimulate them later in the cochlea? Okay, yeah, the high frequency, because their waves are coming in really fast. They're going to hit and distort the basilar membrane much earlier on in the cochlea, where these low frequencies are coming in, and distorting it much later. Okay, so where in the cochlea is displacement taking place? Is it coming early, or is it happening later on? Okay, because your brain is basically mapped out to get information from these cells to a certain location, and these in a certain location, those of medium, okay, in a certain location of the brain. Okay, if these neurons are getting stimulated, the brain can put a, hey, there is high frequency. Or if these are the ones that are getting stimulated, then I can have a low frequency or low pitch. Okay. We did see that these inner hair cells, these are the sensory ones. We did see there are 3 rows of outer hair cells. These are actually motor in that they go to amplify the sounds later in the cochlea, because you got to remember these coming through the cochlea, making these turn. If I have a very low amplitude, I'm going to have a hard time getting that sound all the way up there. So when I get it low frequency, or low amplitude, these can help amplify the signal, so I get it stimulating those outer hair cells later on in the cochlea. Okay, but that would be for quiet, low frequency sounds. I'd have to make it up all the way up there. Okay, but again, pitch, the actual pitch is going to be determined on where in the cochlea is getting stimulated. Okay, higher frequency being earlier on, lower frequency being later on in the cochlea. And we're going to see this mapped out in the next slide, okay, where you can see this. Here's the better picture. You got the high frequency, the red, right? The medium frequency is the green, and the blue is the low frequency. Okay. So you can see high frequency going to be earlier on, medium frequency going to be in the medium, and then the would be, okay, and this is simplified because remember this is going to actually make 3 turns. Okay, but you can see the movement too, scala vestibuli through the scala tympani. Okay, but the earlier on stimulation marks it as a high frequency signal because that's going to stimulate these early on, okay, these earlier parts of the cochlea. And let's see how this relates. And this is where we get the place theory of pitch. Basically, the brain is, this is the auditory cortex, and the information from the sensory cells of the auditory come to, and it has a tonotopic organization. You can see the different regions, the white, the early stretches, then yellow, then red, into pink, and you can see how it is related here. The sensory cells from the white go to this region of the brain. So if these are getting stimulated, remember the earlier, the higher tones, higher frequency, higher pitch are going to stimulate the cells earlier on in the cochlea than those with the lower pitches. Okay, so depending on which sensory cells are signaling, if it's sensory cells from this region, medium pitch, sensory cells from this region are getting stimulated, then low pitch, and then those with high frequency would be high pitch, would be sent to this region. Okay, so that is how we're sending. And again, neural pathways, we get the impulses. They're going to travel to the vestibular cochlear. Okay, but ultimately, where does all good sensory information go to? We already talked about the thalamus. Okay, we get the information to the thalamus, and then the thalamus is going to send that information out to this auditory cortex in the temporal lobe. Okay, depending on which region is getting stimulated, can give us association of pitch. And if they're getting stimulated here and they're getting more bending, they're getting higher frequency of action potential, so it would be a loud, high-pitched sound. If there's a low frequency of action potentials in this region, it would be a quieter, high frequency. Okay, because remember, frequency tells you how strong the signal is. What is the amplitude? Whereas pitch has to do with how frequent the sound waves are. So now let's look at some disorders in hearing, and basically deafness and ability to hear. And it comes in basically 2 forms. We can have conduction or sensory neuron. Basically, conduction is, what do we have to do? Those waves have to be conducted into the external acoustic meatus, to that middle ear, and then ultimately to the inner ear. If there is a disruption in the ability to get the sound waves to the inner ear, that would fall under conduction, ability to conduct. So you could have problems in the outer ear, like your professor. When you go surfing in cold weather, your bone actually grows here in the canal. It can actually shut it off. So that would hinder. You could get some conduction deafness from this. One of the main ways of getting conduction is usually the ossicles here in the middle ear is an air-filled cavity where the ossicles can move. But when you have an middle ear infection, pus accumulates, and these are harder to move, and then they will have less ability to vibrate the sounds on that oval window into the cochlea. So those are some forms of conduction deafness. How do you solve that? That's your normal hearing aid. You can amplify the sound, make it louder to help get the conduction through to the inner ear, to get the sound to. So the problem lies in the conduction. It doesn't lie in the cochlea or the neural area. And that's where sensory neural deafness comes in. Okay. And let me go back to conduction. Like if you fix the ear infection and this gets rid of the pus and stuff, then your hearing comes back. But sensory neural is loss of basically the ability to pick up those movements. So there's damage to the hair cells, the sensory cells. Okay, it can be one part. You could have damage to the nerves that bring the information from the cochlea up to the brain, or you can actually have damage to the areas of the temporal lobe where you get integration of the sounds in the brain. Okay, so that would fall under sensory neural. Until once they're damaged, you can't get sensory neural back. One way to overcome this is if the problem lies in, say, the sensory cells of the ear, those hair cells, okay, but the nerves are still good and the parts of the brain are still good. That's where you've seen if people have those implants, those cochlear implants. Okay, what they do is they vibrate and cause the dendrites to move with sound. Okay, but again, that nerve and the brain's got to be viable, right? If there's any damage to them, then you won't be able to conduct the sounds back, so you get some damage there. Kind of when your mom told you not to listen to loud noises, you can imagine, say these are your little stereocili and your hair cells, right? You listen to loud music, remember the amplitude. Going up here, you can see you could torque these, you can damage these because of this loud sound waves coming in, causing massive torquing to these cells, and you can get damage to them. So there is, when mom said, don't listen to loud music, you're going to lose your hearing. She actually was right in a way. Okay, so, but that would be a form of sensory neural because the damage is happening to the sensory cells. Once these cells are damaged, they don't get replaced, and you would lose the ability, or not the ability, but you would have less ability to hear those sounds. So now let's move into the most complex of the sensory systems here. It's going to involve our eyes that pick up vision or pick up light. Okay. And so what we have is we have photoreceptors in our eyes, back on the back of our eyeballs, called the retina. Right, that's where those photoreceptors lie, and what they are going to transduce is they are going to transduce energy in what we call the electromagnetic spectrum. Okay. You don't need to know exactly the electromagnetic spectrum, but what you need to know is that these different energies travel in waves. Okay? And what the difference is the frequency at which they travel. Some have small wavelengths, right? The distance for a full wavelength And so if it has a small wavelength, it would be much more frequent. Or you can have larger, okay, lower frequencies. The wavelengths are much longer. Okay? And this electromagnetic radiation, this energy, is ultimately what's going to activate our photoreceptors. But you can see it comes in. We have high frequency electromagnetic radiation like gamma and X-rays, ultraviolet, okay, UV lights from the sun. We have infrared, microwave, which are the longer, lower frequency. And in between those, we have this visible light, just a small region, okay, with a wavelength of 4 to 7 nanometers. Okay. This is what your photoreceptors will respond to. Okay. They will respond to electromagnetic radiation within this frequency, okay, within this certain wavelength, because to give us vision. See why when we go to get X-rays is electromagnetic radiation. Why don't we see these X-rays coming down? Why don't we visualize these rays are there? They're going through the air, but the reason is our photoreceptors that we have in our eyes only respond to energy in this frequency, or energy with this wavelength, okay, in the visible light range. The other thing to realize about these wavelengths is, not wavelengths, but this energy is, when it travels through a gas, it can be going in the straight pattern, but when it moves into a liquid, it gets what we call refracted. Okay, because this is what we're going to be doing in the eyeball. We're going to be refracting the light. The light's in the air moving towards our eyeball, and our eyeballs, if you remember, have those vitreous humors. They have those cornea, this different cellular layer. So from going from a gas to a different medium, we're going to get refraction of light, or the electromagnetic radiation. Okay, so keep that in mind when we're talking about the eyeball, because we're going to have these different structures in the eye that allow us to refract the light. Alright, so we do have to do some anatomy here, because we're going to be talking about all these different structures of the eye and kind of their functions. The ones we got to most focus on would be cornea. Remember, that's that clear layer at the front of the eye. Remember, we have the sclera, the white outer area, but in the front, it is this clear layer. That's the cornea. We have the choroid. That's the vascular layer. Okay, lining here. This pink layer with the vessels in it. Okay. And then we have the ciliary body and the iris. Okay. The iris is a muscle. The iris is here. That's the colored portion of your eye, but your colored portion of your eye is a muscle, because what it's going to control is the pupil. The pupil is the hole, the black of the eyeball, because the pupil is where the light that's coming in can actually travel through and ultimately hit. So it'll come through the cornea, through the aqueous humor, remember the fluid that is in here, go through the pupil, and then it will hit this other structure called the lens. Okay, and the lens is controlled by muscles of the ciliary body, this here. So what we can see is we can change the shape of this lens, and then ultimately the light travels through. We have the vitreous humor in the posterior chamber of the eye. Okay, it is not air. Remember, it is gelatin-like here, aqueous up in the front, and then we have on the back here, we have the retina, and this is where we have our photoreceptors. Okay. You see, we'll talk about this little specialized region, this fovea centralis. It is a part, just a small area of the retina. But what we're going to be trying to do, what your eye tries to do, is to focus that electromagnetic radiation of visible light to this fovea centralis. Okay, because this is where we have our receptors, and in our fovea centralis we have a very high density of receptors. So if you want to have good vision, you want to get the light focused at the fovea centralis. So just looking at a close-up of this, you see here's a ciliary body. This is a muscle, circular type muscle. So circular, right? When it contracts, it's going to shorten the circle. When it relaxes, it's going to open up. Okay. And we have the anterior chamber here. That's going to have our aqueous humor, and behind the lens, our posterior chamber, we have the vitreous humor. Okay. The other muscle we have here is the iris. Okay, that's the colored portion of your eyes. If you have green eyes, this would be green in color. Okay, but it is a muscle because it's going to control that opening here. That opening that would be here would be the pupil. Okay. All right, so what we're going to see here, we're going to look at the iris here in a second, but to get good vision, to get good, solid, crisp vision, we have to control a few things. We have to control, we saw the refraction of the light. We have to get the light, or the light energy, focused to a certain point in the back of the eye. The other part that we have to control is we have to control how much light is coming in. Because if it's very bright out, we don't want to let a lot of light in, because it actually will bleach out our receptors, our photoreceptors. Get bleached out, and it will be hard to see visually. That's why when you look at a bright light, you start to squint and you'll see your pupils. Okay? Your pupils will go from big or larger to a smaller diameter to can limit the light. Okay, so that is one factor. We got to limit the light, and we also have to control the refraction of that energy. So our iris is going to be involved in controlling our pupil diameter, how wide open our pupil is. Okay, and you can see our iris is actually made up of two muscles. There is this inner circular muscle, okay, and this outer radiate muscle. This is smooth muscle, so it is. We haven't talked autonomic, but smooth muscle is controlled by the autonomic nervous system. Okay, autonomic means I don't have, it is involuntary. Okay, I don't have to sit there when I go out in the sun, walk out from a building and walk out in the sun. I don't have to say, all right, I got to control my pupil diameter. It is done automatically for me by the autonomic nervous system. Okay, but you can see in dim light. In dim light, we want to have more light coming in. We have very little light out in the environment. We want to have as much coming in so we can stimulate those photoreceptors. Okay, and in that case, the muscle that will be getting contracted would be this radiate. You can see if this muscle shortens, if these muscles shorten that are radiating this way, it's going to pull the pupil open. So this has to contract, and that circular muscle has to relax. Okay. And this is actually controlled by the sympathetic nervous system. Okay, the fight or flight. We're going to talk, our next lecture is going to be on this, so just keep this in mind. This is controlled by the sympathetic, this muscle. Okay. In bright light, okay, now we want to limit the light coming in, because otherwise we're going to get our photoreceptors bleached. So how do we do that? Now we can relax this muscles, the radiant muscles, and contract the circular muscle here, and that's going to cause our pupil to constrict, okay, to limit the light coming in. Right? And then in normal light, you can see we're kind of in between these two states. Okay? But this circular muscle is controlled by the parasympathetic. Okay? These two work antagonistically. So usually when one is working, the other is dampened down. Okay? But we're going to see this in the next lecture when we do autonomic. Okay? We'll get into muscarinic antagonists when we get to autonomic nervous system, because what we're saying is the receptor that controls the contraction of this muscle is a muscarinic receptor. Okay? Acetylcholine's being released, and there are muscarinic receptors on the muscles in the circular. If I cannot contract this, then what's going to happen? My eye is going to stay dilated. So if I have an antagonist, a muscarinic antagonist, I can keep this muscle from contracting, and I would have dilation. But we'll touch on this more when we get to the autonomic nervous system. But realize what the iris's function is to control the amount of light coming in. Okay, in dim light, we want to have more light getting in to those photoreceptors. Okay, so we dilate it. In bright light, we don't want as much light in because all this light would bleach out the receptors and we wouldn't be able to make any distinction between light, and so we want to constrict. So now we've limited the light coming in. Now we got to refract the light, and then ultimately I need to add one third component, is we have to make sure our eyeballs are looking in the right direction. Okay, so we're going to need three main factors. We got to limit the light, okay, depending on whether dim or bright. We're going to have to refract properly because we have to get refraction back to where those photoreceptors are located in the back of the eye. And we are also going to have to get the eyeball to move in the direction that we want to look at. Okay, so we need those three things. If we have a problem with one of those three things, we're going to have some disruption in the vision that we see. Okay, so the iris, we already controlled the light. Now we have to do refraction. Let's bend the light coming in to get it focused in the right location in the back of the eye. Because if this light comes in how it is, it won't be focused where we need it to get to stimulate those photoreceptors. Okay, there are three main components: the cornea, the lens, and the humors. Okay, those fluids inside the eyeball. The one that is going to be doing the majority of the bending is going to be the cornea. Okay, that outer clear portion of the eye. Right? But what we're going to see is light coming in from far, looking here, and light coming in from shorter are coming in at different directions or different distances away from each other. So this one, you can see we're going to have to focus. Okay, we're going to have to bend it much quicker to get it focused in this location back here. Where light coming from afar, we don't have to bend it as abruptly. Okay, you see the distance between the rays here, where here it is greater. So we have to bend close objects much quicker. So what we're going to have to do is we have to change the refraction of the light coming in, depending on if it's coming from far or if it's coming from close. Right? And that basically is the function of the lens. Because what you see with the cornea does not change shape. Right? So as a lens, it is not going to adjust. How it is arranged, it's going to bend how it bends because it doesn't change shape. The way we are able to change the amount of bending of the light is going to be through the lens. Okay, because the lens, if you remember the ciliary body here, that muscle, and these little suspensory ligaments connecting to the lens, we can either stretch the lens out, okay, or it can be more round, bubblegum shape. Okay, this would bend the light less, this would bend it, refract it more. Okay, so depending on what we're looking, the lens can change shape to alter the refraction so we do get the focal point for the light at this location in the back of the eye. Lastly, the humors, okay, they don't change at all and they do very little bending, but they do some of the bending. But the two main players are going to be the cornea and the lens. The cornea is going to do the bulk of the refraction, but the lens is going to be able to alter the refraction. So let's take a look at that lens. Okay, and you can see it has ciliary muscles here attached to the lens by these little suspensory ligaments. Okay, and remember this muscle is circular. So when the muscle contracts, it actually would be smaller, or say the lens is in here. Okay, these ligaments here. The lens is flexible. Okay, it is elastic. It can be stretched, but then it bounces back. Okay, so it is elastic in shape or in structure. Okay, so we can change the shape of it. We can, when the ciliary muscles are relaxed, it's more open and those suspensory ligaments are pulling on the lens and stretching it out. Okay, because the muscle is farther away, the suspensory ligaments here are pulling on it, stretching it. When the ciliary muscle contracts, right, now we're in this shape. Now there is less pull by the suspensory ligaments because the muscle is moving closer to the lens when it contracts, and the lens that is elastic comes back into this kind of gumball shape, I like to call it. Okay. So this shape, this more rounded shape, can bend the light waves much quicker. So when we need to do that is light waves coming in from close. Okay, they are farther apart. See the distance here between the distance from afar. These are less distant, and therefore we have to bend less in the distance. So in close range, near vision, right, we have to bend the rays much quicker. So we have to contract the ciliary muscle, relax it, and then the lens will, because it is elastic, will come back into this more rounded shape. Okay. In stuff looking from far, distant vision, right, we have to bend the light waves less, and therefore when the ciliary muscle relaxes, it's more stretch, and this form, this shape of the lens will bend the light less. Okay, if we kept it round, this would actually be too far out. Okay, we would bend too much. This is all good and great when you're young because this elasticity of your lens is nice and supple. It can bounce back into the shape. But what happens when you get old? I like to tell my kids we get old and crusty, and that's basically what happens with the lens. It gets older and crusty, and when I go to contract, the elasticity of this over many years has decreased, so it can't get this round. And so if it's still stretched out more, it can't bend as quickly, and therefore you lose your close vision. Okay, that loss of flexibility with age is called presbyopia. That's when you need those reading glasses. Even if you have good vision, okay, you're going to lose some elasticity, and ultimately depends on how much this changes, but in the last 2 or 3 years, mine has changed drastically because now I got to use reading glasses because my lens can't get back to this shape. Okay, when it gets relaxed, it doesn't bounce back. It's not as elastic as when I was a young pup. Right. So now let's look at some disorders in the ability to refract. Okay, we can get myopic eye, which is nearsighted. You can see near but have trouble seeing distant vision. And hyperopic, where you're farsighted. You can see closely, but you can see from afar, but you can't see stuff closely. Okay, it can come of two things. We can have, but you can see what happens. We want to focus the energy back here. This is the fovea centralis. Okay, fovea centralis, this is where we have high light energy. Okay, here we get proper focusing. We're getting the energy, the light being focused right where it needs to be, and same here. But where we're focusing is the fovea centralis, because this is going to be the region in which we have the highest density of photoreceptors. Okay, so this is what your eye is trying to refract. It's trying to refract it back to this region, fovea centralis, because that's where we're in our. If you have more receptors, you're able to get. Remember our 2-point threshold. If you had lots of density receptors, you were able to distinguish 2 points much closer together. That's the same with the fovea centralis. There's higher density, so we're able to distinguish between different light energies much better at this fovea centralis. Okay, so we can get this nice, crisp, clear vision. So let's look at the myopic eye and nearsighted. Okay, the lens could be too strong or the eyeball too long, and what this ends up with is, because a normal lens can only change its shape to a certain degree. If the eyeball is too long, the lens may not be able to compensate to bring the focal point back here. In a long eye, the focal point ends up short of the fovea, and you can see the rays are sprayed out. Me, I have very bad vision, and so when I go take my glasses off and look at a street light, that street light almost looks like those green light almost looks like a big Christmas ball. Okay, the light spraying out, it looks like it's 3 feet big because the light is all sprayed out. It's not focused and not crisp. Okay, the light waves are sprayed out there. Okay, how do we correct for this? Right, you can use a concave lens. Okay, what this does is spread out the light as it comes in, and now even with the long eyeball, the light can get focused at the right location. Okay, with a hyperopic eye, we got the opposite. Eyeball could be too short or the lens too weak, and what happens is the light doesn't get focused soon enough. The focal point actually falls behind the retina, behind the fovea centralis. So in that case, we want a convex lens that will cause the light to be bended more before it hits the cornea, and therefore we can get the focal point moved up. in that shape. Okay. So myopic, nearsighted, hyperopic, farsighted, and then that change in elasticity when you get old is presbyopia. All right, so now let's look at where we're actually sending that light. We're sending that light to the retina because that is the region in which we have the photoreceptors. That's part of the eye, the back of the eye, in which the photoreceptors lie. But there are two layers. You can see light. It's coming in this way, in this direction. And there is a pigmented layer and a neural layer. Okay, the light is actually going to pass through the neural layer first, right? And then you can see the pigmented layer is here. But in the neural layer, you have basically three main cells. You have the ganglia cells, these are neurons, bipolar cells, okay, and then you have the photoreceptors. Okay, these are the receptors. These are where the light is actually going to get absorbed, and we're going to get stimulation of these photoreceptors to cause signaling through this direction. Okay, but what's interesting about this is you can see the light actually has to pass through this neural layer before it gets to the actual receptors. The receptors aren't out here, okay, and then the signaling comes backwards. The light has to pass through, and then the signaling happens in the opposite direction. So let's look a little bit closer at the neural layer. Okay, three distinct layers to it. We have the ganglion cells, bipolar cells, and then the photoreceptor. These are the actual receptors that are going to be stimulated by the light energy. Okay. So the photoreceptors, there are two types of photoreceptors. There are the rods. Okay. They are more abundant. That is these here, yellow, and then the cones, more cone-shaped, are the blue. What you see are rods are very sensitive to light. So you can have low when you're in dim light, say like at dusk. The rods are more sensitive because, or can pick this light energy up because they are more sensitive. You don't have to have as much light to stimulate them. Okay, so we use these in the dim light. But what you lose is you lose acuity, you lose crispness or clearness of vision. The cones are less sensitive to light, so you need bright light, brighter light to stimulate the cones. Okay, and the cones are what are actually going to give us our color vision. And we're going to see there are three colors: red, blue, green cones. Right, but these are where we're gonna, the light is coming in and going to stimulate these photoreceptors, and then the information, the neural pathway, is sent from photoreceptors in the opposite direction. First to the bipolar cells and then to the ganglion cells. You see the ganglion cells, their axons are what merge to ultimately make those optic nerves. Okay, the optic tract that is going to take the information towards the brain. So a closer look at the actual photoreceptors. See, here are the rods, here is the cones, and you can see there are two there's an inner segment closer to the eyeball, and then an outer segment with these disc like regions. Okay, the inner segment, it's like the normal cell. It's going to contain your organelles, your nucleus, and so forth. And we're going to have the synaptic ends on this end. But the outer segment has these flattened discs. right? And this is what's going to have these photopigments. These pigments in here are what are going to trap or capture the light to stimulate these photoreceptors. And what we're going to see is there are different pigments to pick up different wavelengths of light. Remember, we had those different wavelengths of light, and we're going to have different pigments to pick up different wavelengths of light to stimulate the cells. Okay, and when we see this outer segment is embedded in that other layer, that pigmented layer, and we'll see these are closely related with the photoreceptors, the rods and cones. Okay, because this pigmented layer, kind of like we saw the astrocytes for the brain, they're kind of like these cells of the pigment layer are kind of the assistants to the photoreceptors here. They phagocytose the photoreceptors, so they phagocytose these discs, and these discs are continually replaced, the ones that have the photopigments. There's also melanin, therefore pigmented. Melanin is a pigment, and what it's going to do is it's going to pick up the extraneous light, so the light that actually gets absorbed here can give us a clearer picture. They also supply the nutrients, okay, suppress immune attack, and they're also involved in recycling the pigments that are used to trap light here. Okay, so you can see all the functions here, kind of like the personal assistants to these photoreceptors, because basically they're going to allow them to operate and function properly. So let's first focus on the rods. Okay, remember these outer segments, they have these disc region with these pigments. In a rod, the pigment molecule is called rhodopsin. Right? And pigments absorb light. In this case, absorbing light roughly around 500 light with a wavelength of 500 nanometers. Okay? And you can see where it falls on the visual spectrum. It falls between blue and green, closer to green, right? So if you see at night, when it's starting to get dusk out, that is when you start using your rods, because remember, these are more sensitive to low light. We can use them in low light, where the cones we have to use in bright light, or we need bright light to be able to stimulate them. But rods we can stimulate with dim light. And if you were, say you saw a hit and run and the car was red, you would have a less of a chance to get it right unless the car was green, because as rods fall close to the green structure, you're able to see the color green more easily at night. Okay, but what do you see with rods is, even though they are more sensitive to light, we lose acuity. So when it starts getting dusk out, how does it feel? Does it feel like you have really crisp vision, or does it feel kind of fuzzy? It feels kind of fuzzy because you lose acuity when you need to use the rods, but these allow you to see with that dim light. All right, here's where vision gets fun. You got to think of this in reverse, because what have we seen with most of our our receptors, is that activation or the stimulus causes them to depolarize and to send neurotransmitter, or to be stimulated, depolarized. All right. In the case of these photoreceptors, what we see is in the dark, so lack of light, lack of stimulus, we are going to see that the photoreceptors are being depolarized and sending neurotransmitter. right? That's opposite, right? Because you expect light to hit the photoreceptor and cause it to depolarize, and then the signaling starts. But that is not the case in the photoreceptors. In the dark, we get depolarization. What we see in the dark is, here is our pigment that absorbs the light. Okay, in the pigment there is this retinol molecule. Okay. In the dark, this retinol molecule is 11 cis. It is in the cis configuration. Okay, and this is associated with these G proteins. Right? In the dark, in the cis, the G proteins are not activated. Okay. And there is an enzyme, phosphodiesterase, is inactive. Okay, because to get this enzyme active, I need to have the G proteins activated to cause phosphodiesterase. Okay. What phosphodiesterase does is, you can see, it takes cyclic GMP and turns it into GMP. Okay, without phosphodiesterase being active, we're not converting cyclic GMP. So there is a lot of cyclic GMP. And cyclic GMP binds to a channel, a sodium channel, to keep the sodium channel open. Okay, this is what we call the dark current. The sodium moving in when it is dark. If sodium is allowed to move in, what's happening is this cell is being depolarized, right? Depolarization. What does depolarization cause? It usually causes the cell to be depolarized, causes release of neurotransmitter. Okay, so when in dark, the rhodopsin is in the cis configuration. The cis does not activate the G proteins, and the G proteins don't activate the phosphodiesterase, and so cyclic GMP levels are high, keeping the sodium channel open. Okay. When light, the right wavelength of light, hits rhodopsin, okay, remember it's about 490, around 500 nanometers. When that light energy hits the rhodopsin, this retinol molecule gets converted from cis to all trans. Okay. And that will cause activation of these G proteins. Okay. Now the G protein goes and turns on that enzyme, phosphodiesterase. And phosphodiesterase converts cyclic GMP to GMP. Now we are decreasing the levels of cyclic GMP. Remember, the cyclic GMP over here was keeping the sodium channel open. Now, with low cyclic GMP, we're closing the sodium channel. Sodium can't rush in, and what will happen is the cell gets hyperpolarized, okay, or repolarized. In a hyperpolarized cell, we're not releasing neurotransmitter, and we're not talking to those bipolar, because remember, the photoreceptors talk to the bipolar cells. So again, somewhat backwards here. So what is happening, here's the dark, and we'll walk through it again just because I know it is a bit confusing with the reversal of how these cells are stimulated. Okay, but in the dark, cyclic GMP levels are high. There is the phosphodiesterase enzyme is not turned on, right? And those sodium channels open, and we're depolarizing the cell. The depolarized cell causes release of neurotransmitter. And here's where it lies, is the neurotransmitter that is released is inhibitory. So the signaling is photoreceptors talk to the bipolar cells, and then the bipolar neurons talk to the ganglion cells, and then that information is brought to the brain. Okay. In the dark, because these are depolarized, we're releasing inhibitory neurotransmitter, and that prevents the signaling of the bipolar cells and the ganglion cells. Okay. Now in light, okay, we hit rhodopsin, we go from cis to all trans configuration for retinol, and we turn on phosphodiesterase, and phosphodiesterase converts cyclic GMP to GMP. That will result in closing of the sodium channel. Now the photoreceptor is hypopolarized. Now it is not sending that inhibitory neurotransmitter. Now these bipolar cells are free to signal to the ganglion cells, and then the ganglion cells will ultimately send that information back to the brain. Okay, so in dark, photoreceptors are depolarized. In light, photoreceptors are hyperpolarized. What the difference is the neurotransmitter. We saw with the other sensory cells is when these cells got activated, they sent excitatory neurotransmitter. In this case, the photoreceptors are sending inhibitory when the stimulus is not there. When the stimulus is there in the form of light, we stop the release of inhibitory neurotransmitter. All right, so now with the cones, that was with the rods, we had rhodopsin. Okay. In this case, the cones contain a pigment called photoopsin. And three different types is we have cones that are called S cones, or short wavelength, S for short wavelength, which are the blue cones. We have the M, or medium wavelength, our green cones, and then we have our L, long wavelength, or red cones. Okay, so this is what we call trichromatic color vision. We have three different colors in which we put together to give us our different colors. Okay, but the signaling is very similar. It's just the pigment, instead of rhodopsin, which we saw on the rods, it's going to be photoopsin. And each cone is going to have a different photoopsin picking up the different wavelengths. Okay, depending on which cone it is, depends on which photoopsin is there. The thing with the cones is we need bright light. So these are less light sensitive. We need brighter light to stimulate the photoreceptors, but what we saw with the rods is we got low acuity, right? In the dusk time, we have very fuzzy vision. It's not very crisp. But if we have bright light, okay, we can stimulate the cones, and that will give us our crisp color vision. So here is a slide that I just added, so I'll put this on onto Canvas, but usually this is covered in the lab, but we'll cover it here in the tricolor vision. We have the cones with the color vision. We have the three different cones. We have the blue, green, and red, okay, with the short wavelength, the medium wavelength, and the long wavelengths. Okay, and so how are we going to get all these different colors? We only have three colors, or for our photoreceptors. We got the blue, the green, and the red. And so what the brain does is it puts perceptions to the different signaling. You can see if we get a combination of red cone signaling along with green cones signaling, the brain perceives that, puts a perception as a yellow color. Okay? Whereas if we get a combination of blue signaling with almost equal signaling from green, then we get this cyan, this light blue color. Okay, and you can see there's many variations. You know, we can have, when we have white, we're actually getting red, equal amount of red, blue, and green, all three colors, and the brain perceives that as white. Okay, and what the brain is actually perceiving is the reflection of the electromagnetic radiation. Okay, for something that's red, it's reflecting the red, but what it will absorb the, so like a red blouse will absorb the green and the blue light energies, but reflect the red. So what the light is perceiving is a reflection of the electromagnetic radiation, in this case, in the wavelength of red. Okay, and so that is how the perception is perceived. The brain is going to receive signals from these three different colors, or there's three different photoreceptors, and whatever signals it's receiving, it can put a perception on what color it is seeing. As we said, we get our crisp vision with our cones. And so we have regions in the retina, that back portion of the eye, where we have highly pigmented or high area of cones, right? And we have this larger area, this macula lutea. It's a high density cone, and right in the smack dab in the middle of that macula lutea is the fovea centralis. Okay, we got this little dot here. Okay, and this is where your eye muscles work to make sure that the lights are focused here, your refraction is focused here, and then your iris, okay, regulates the amount of light coming in, so we get a nice focus at this fovea centralis. Okay, and this is where we're going to get our sharpest vision, because we have our highest density of cones, and cones give us that high acuity. Okay, so this is where we want to focus the light, in that little pinpoint region sitting in this larger macula lutea. You probably have heard the term macular degeneration, and that essentially is degeneration of that macula lutea region. Okay, and what you see is somebody with normal vision, if you focus on the dot, okay, you see all the grid lines, or if you're focusing at the picture, you're seeing a nice clear vision. But somebody with macular degeneration, okay, because remember, your eye is always going to focus your eyeball, okay, wherever you're focusing, it's going to try to put that dot at the fovea centralis, in front of them, and where that macula lutea, inside that macula lutea. So your eye is always going to focus there, so you can see you have basically a hole where there are no photoreceptors that have been degraded. But again, your eye is going to focus at the fovea centralis, so that box wouldn't be over here. Okay, it's right in the center where your eye is trained to focus, because there is high density of receptors there. Okay. And what we see, why do we get such high acuity for visual acuity there, also is we have high amounts of cones in the fovea centralis, but also you can see kind of this indentation where the light has to come through all those layers. But at the fovea centralis, we almost get like a parting of the neurons, so the light is able to focus more directly on the cones that are located in the fovea. Also, what we see is there's no convergence. Okay, we have 120 million rods, 6 million cones, and we only have 1.2 million axons that are leaving to make up, okay, that optic nerve for each eye. That means there are multiple photoreceptors ultimately converging onto one neuron that's going to send the information back to the brain, right? And what we see in the fovea is one photoreceptor to one ganglion cell. So we don't have convergence onto the ganglion cells. Okay, one photoreceptor is going to send one axon to the brain in the fovea, in the cones of the fovea. Okay, and therefore, just kind of like that tactile, right, where we had high density receptors, we were able to distinguish 2 points. Here in the fovea, we're able to distinguish between 2 forms of light energy much more readily because of this setup with low convergence and the parting to allow the light to come in. So here's a good visual representation of that convergence. You can see here's our rods, and ultimately all the rods located along here are going to converge onto one ganglion cell. This is what sends the information to the brain, okay, or headed in that direction. You can see the visual field is much larger. So any light sitting here is only going to stimulate, even though it might stimulate these different photoreceptors, it's going to stimulate the same ganglion. Where here, light energy that hits here will be sent by this ganglion to the brain. Light energy that hit here will be sent by this one. And so you see the receptive fields are much smaller, and therefore we can get much more distinguishing or crisper vision. Okay, that light that's coming in here is ultimately just going to stimulate this one, and so it will only stimulate here and not these other photoreceptors. Okay, because low convergence, we're able to get crisper vision with the cones that are sitting in that fovea. So you can see for the neural pathways, we have 2 different pathways coming through here. We have the geniculostriate system. That's going to tell you, that's going to be the system to give you a what are you looking at. I'll put reference to what. And the tactile, is the where pathways. But let's look at the visual fields and what side of the brain the information goes to. Okay, if you remember for cutaneous, information on the right side goes to the left side of the brain. Now that's not the case with the eye. All the vision, all the light energy coming into, this is looking up from the bottom. The left eye doesn't all go to the right side. Everything from the right eye doesn't go to the left side. What it is everything from the left visual field, is going to pass to the right side of the brain. You see the yellow. And everything in the right visual field is going to go to the left side of the brain. As you can see, the light coming in to the left eye, or from the left visual field, is going to hit the left eye in the medial location, where that is going to hit the lateral in the right eye. Okay, so the medial from the left and the lateral from the right end up going to the right side of the brain. All right, and then the opposite. From the right visual field, you got the light's going to hit the medial side of the right eye and the lateral side of the left eye, and you see both of those information from the right visual field head off towards the left side of the brain. Okay, and as they go back, again, we have the geniculostriate region here. These are going to be the what pathways, and then the tectal is going to be the where pathway. You see the tectal, the where, and then this is the what pathway. And there we have it. That is it for vision. I know it's a long haul, but we are done with sensory, and now we can move on to autonomic, which is fairly more simpler than the complexities here that we see in the sensory.